Hi everyone, we're back. Video 3 of 4 in the Cytoskeleton and Intracellular Transport Lecture 30. Here we're talking about how muscles contract, and you'll see that there are a lot of parallels with how motor proteins travel along microtubules that we did in Video 2. The big parallels have to do with ATP binding, ATP hydrolysis, and changing conformation. Here we're going to look at another one of the cytoskeleton types of filaments. We talked about microtubules, microfilaments, and intermediate filaments. Here, microfilaments are a part of muscle contraction. Microfilaments are made up of actin. Actin and myosin are the two main proteins that function in muscle contraction. Actins make up the microfilaments in the body. They do more than just provide for muscle contraction, but we'll see them used here in muscle contraction. So again, this is actin monomers bound together in a polymer, a long chain, and then a microfilament is just a double helix of two long chains wrapping around one another. Here are three types of muscles. Actin is involved in each. We're going to look at skeletal muscles specifically. These are a little different in function than some of the other muscles in the body. Skeletal muscles have remarkable range of moving along long lengths. So just think about our legs when we're running, or our arms when we're doing a curl. We've got a long distance of movement, and skeletal muscles are adapted to do that. We're not exactly sure how they travel or contract along those long lengths. That's an area of actual ongoing research. Here is a picture of those skeletal muscles, and you can see that we're looking at more and more detail when we go from muscle all the way down to the sarcomeres. So there are bundles of muscle fibers. Muscle cells are actually very large and have multiple nuclei. Within those cells are these myofibrils. Myofibrils are consecutive sarcomeres. So a sarcomere goes from Z line to Z line. And within sarcomere are both actin and myosin filaments. So the actin are shown here as the tan lines and the myosin as the purple lines. And we'll see those in more detail right away. Sarcomeres contain thick and thin filaments. So actin molecules are the thin filaments. They are surrounded by another protein called tropomyosin. That's the long green strand here. And we'll come back to that in the next video. But the thin filament is made up of the actin polymers and tropomyosin wrapped around it. The other element of the sarcomere is the myosin. And so myosin, again, different protein than actin, forms this double helix, two chains wrapped around one another. And notice they have these heads. So the head is kind of off the screen up here, but it's shown in the picture in the bottom. And those heads are what binds to actin when our muscles contract. All right? So thin filament, thick filament. Sarcomeres have these alternating thick and thin filaments. So the Z lines define the ends of the sarcomeres. And you see how the actin, kind of the tan or red here, how those monofilaments are interspersed with the myosin filaments. So this is a longitudinal section here showing the thick and thin filaments. There's a nice electron micrograph of the same down at the bottom. Here's an end-on version looking at the alternating thick and thin filaments as well. Our model, and remember a model is something that we have lots of experimental evidence for, but we haven't maybe codified that or made it as formal as a theory. So our model for how muscles contract is the sliding filament model. And the idea is that when a muscle is relaxed, there's some overlap of myosin and actin here, the different filaments. But look what happens when you go to contraction. There's a lot of overlap. So it's that the Z discs don't, or the Z discs actually move a little closer together. It's because the myosin moves along the actin and pulls those Z discs together. That's what contraction is all about. All right. So myosin is just going to move along the actin, pulling the actin filaments toward the center of the sarcomere. Okay. So that's the sliding filament model. They slide along one another. The thick filament really stays in a sense, but it moves along. More appropriately, it pulls the actin along the myosin. So actin, like I said, moves toward the center. All right, so it's clarified down here in these 3 bullet points. Thick filament stays in place. Myosin heads walk along the actin. So the thick filament myosin heads walk along the actin, and this pulls the actin inward. The cross-bridge cycle is part of that model. This is how things slide. So let's start at the top with step 1. The myosin head binds ATP. No surprise there, right? We knew that was going to be part of it. This is the low energy configuration. ATP has come on, it binds myosin, and you see the 2 filaments are separated from one another, the thin filament and the thick filament. Step 2, what happens? Well, we can pretty much guess ATP is going to be hydrolyzed. And sure enough, it is. ADP and phosphate phosphate is separated from ATP to make ADP. They are still bound to the myosin head, but that changes the conformation. All right? Again, chemical change, hydrolysis of ATP, changes the conformation. The myosin is now in its high energy conformation. So in step 3, step 2 really being the hydrolysis, step 3, ADP and PI are still bound. The myosin head then now moves to bind the actin filament. All right, so the hydrolysis of ATP in step 2 triggers the buildup of myosin. It kind of cocks it in a sense where it's ready to spring forward. And here it is sprung forward and it's binding to the actin filament. Then ADP in step 4, ADP and PI are released. What happens when we release a molecule or bind a molecule? Typically, conformation changes. So sure enough, the position of the myosin changes. So where you can see kind of the angle here of the myosin, I'm down here at the bottom. The angle of the myosin, say 45 degrees. Here, look how steep that angle is. The myosin is actually moved. And that's what tugs the thin filament toward the center of the sarcomere. So, step 5, ATP binds again and we start over. Low energy myosin conformation, hydrolyze the ATP, that cocks the myosin head and it binds to the actin. You release ADP and PI, you get another conformation change that actually moves the myosin head and the actin filament is slid toward the center of the sarcomere. So those are the sliding filaments. So the cross-bridge cycle is key. So there's the full, I filled in the red wording to explain the full cycle. There are two videos, or one animation, one video to watch, and then we're done with this. I'm done talking, but please go watch the animation on Canvas about myosin movement. And then go watch the video on YouTube, which is a summary of muscle contraction. So it's a fun video. It's, I would suggest you start at 3 minutes and 27 seconds as it shows up here at the top, and watch it basically to the end. Okay, watch it as long as you want, but it's a good look and nicely done, and it's a fun video about how muscles contract. That's it. Thanks for watching video 3. We've got one more to finish off the story of muscle contraction.